A second part here. Then I got to drink of water. And again, here's some just some tools that molecular biologists have used over the years to identify the organization of the cis regulatory region. Yeah. So we're just going to learn 5, and the first one is DNA deletion studies. People refer to them as promoter deletion studies because we know these good modern terms for a gene being 2 parts: the transcription unit and the cis regulatory region. It really features a promoter proximal to right next to the transcription unit, and it's usually just going to help the RNA polymerase land, and up here an enhancer. So let's say we want to use DNA deletion studies because we don't know how that myogenin gene works. So this would be the myogenin gene. It could be any gene. People still do this a little bit today, but it's really somewhat of a historic thing. But why would molecular biologists talk about it? Because it's physical, and the methods show the physicality of the work. And so what it's like is this, is we just start out with this long piece of DNA in a test tube. And what we're going to do is we're going to start at this end, and we're just going to start removing DNA like that. And to do that, we're going to use an enzyme that degrades DNA, and it's called a nuclease. And it's called a DNA nuclease because there are also RNA nucleases. And they're just found in every cell. You need them every cell needs them to metabolize, to break down DNA or RNA when you don't need it. And the kind of DNA we're going to use is called an exonuclease. And an exonuclease, it's one word actually, exonuclease. And an exonuclease starts to cut at the end of the DNA. That's different than an endonuclease that cuts anywhere in the sort of the interior, that clips it like a scissors. So the exonuclease you could think of as chewing on the end of a piece of spaghetti, and the endonuclease as being like scissors that just cuts randomly anywhere along the length of the DNA. So we definitely want to use an exonuclease here, and we just we do a first reaction when it goes for just a short minute or two, and we get a shortened piece of DNA, and we put that into a cell, and we ask, can MyoD still activate the myogenin gene? We don't know about MyoD yet, but can the muscle cell, still activate this gene? And we know from our modern understanding that MyoD could still bind there, and it would activate the gene. So we get activation of this gene in a test tube. And we're just trying to map where is that activating part of the DNA. So then we do another experiment. We let the exonuclease work a little longer. And of course, blah, blah, blah. And it does. And we still get activation. And we get a little bit longer. And yep, we still get activation. So it's still working. And the enhancer cannot be in this region because we chewed it up. And then the very next one, no activation. So that's another reason why people call this the enhancer, and why they really focus on it, because in these early DNA deletion studies, they got to a place where there is no more activation. And then you would say there's a part of the DNA that starts right there. And we don't know how long it is at this moment, but we do know that it's really important right there. Okay? And that's what DNA deletion studies are. And you just need to explain them. And each of these could be a 2-point question on a test. What is a DNA deletion study used for? Mapping enhancers, and how does it work? Use an exonuclease and chew in from the end, and just keep chewing until the target gene no longer works in a transcription assay. Okay, so that's the first one. Yay! And I might just be able to do all these here in this short second part of today's snow day lecture, because the next one is nuclease footprinting, and in this one we're going to use an endonuclease. Okay, and what it's going to be like is we'll take that same piece of DNA, and what we'll want to do is we'll kind of represent that same piece of DNA in a test tube, many copies of it. And it's that myogenin gene DNA, so we have the transcription unit, the promoter, and the enhancer, and we're trying to map where the important parts of the regulatory region are. And we're going to use an endonuclease, so we're going to get random cuts in different pieces of DNA in random locations like that. Just random, just a short incubation with an endonuclease. And what we're going to do here to help us with this experiment is we're going to have already labeled the end of the DNA with some radioactivity. Okay? And so, so after this short incubation of the radioactively labeled DNA, so a short incubation with the endonuclease, we can do a gel. And so we can do gel electrophoresis. And when we do, the radioactivity will be in different lengths, of all the different lengths possible along the gel, like that. And people call that a ladder because it looks like the rungs on a ladder, and we just see all of them. Now, if we do it with no proteins first, we would get cuts randomly everywhere along the length. We could have lots of cuts right here, but see, we only see the radioactive labeled fragments. So see how we see pieces that are short or longer. Doesn't matter if it cuts in multiple places, we just see this one radioactive piece. And that's the purpose for the radioactive tag, is we're only looking in from this end. Now, look at this. We would see the full ladder on the gel. Now, if we just add proteins in a second set of experiments and now do the nuclease footprinting, what we'd see is we'd see fragments this length, but we'd never see a fragment this length. We would see fragments this length, like that. And we would be able to see the promoter in this experiment because we'd see a fragment this length, and this length, and this length, but we'd never see fragments that length because we're just labeling from the end, and we'd see pieces that are as long as that or longer. But what's happening here is the proteins like MyoD and the pre-initiation complex, they protect the DNA from getting cut by the endonuclease. It can't get it. So we get what's called a footprint. We get a gap here in the gel where these lengths are missing. These lengths that are this long are present. These fragments that are this long are present. But the ones that are exactly that length, and however wide that is, are missing. So we get a gap or a footprint. And that's a nice name for it because of that. And people used to call these protection studies also. Yep. So I think I have some slides of that. Oh, and so, the way you work the thing is you're going to do an extract to get the proteins from the nucleus and use those in your experiments. And, you isolate the nuclei and break them open, and that's get your source of proteins. And so here's the footprint protection assay, and you use the radioactive label. And here's the first, or here's the whole experiment where we have the protein and it's protecting the fragments. So it's protecting fragments that are this long. So the longer fragments are there and the shorter fragments are there, but there are no fragments that are this length. So we're missing some fragments here. And that's the product, and that's what it looks like on a gel. So this is what's actually in the test tube, and then that's what's on the gel. And this just shows another view of it from another figure in a book, and it just shows that these were the parts that were protected by the presence of what? The MyoD protein in one region shows exactly where the enhancer is. Remember, in the first DNA deletion studies, we went in from the end, and we could just see where the start of the enhancer is. Here we get to see the size of the enhancer, and they're very short, only about 20 nucleotides long. Yay. And we also get to see further on, and we see other enhancers, maybe, or, because even if they're not active, they show up on this footprint. For example, we just learned in the first part today about GAGA factor, and we would be able to find where GAGA factor binds, even though it's not really regulating transcription. The deletion studies only are done in ways where you're going to assay whether the gene is still active. This is not about gene activity. It's just about physically where proteins are bound. Yep. All right. So then gel mobility shift assay is another really straightforward strategy. And what that's like is we're going to start out and we're going to just run a gel. And in this case, we'll run a gel and we're going to run it just with a specific piece of DNA. And so we could take the myogenin gene and we could say, okay, so we've done some deletion studies and we found that this is the start of the enhancer. So that's the first strategy. And then the second strategy would be able to map. This is actually the enhancer. So nuclease footprinting shows us that that's the region that gets protected. And so the gel mobility shift assay is to actually just take this piece of DNA. So again, these are always DNA. We take this piece of DNA, probably take a little bit longer piece of DNA, and we just can take that DNA and we could radioactively label just so we can see it, and we just run it in a gel. And we had set it running to that fast, right? Just with DNA. Then over here, we run another sample, and in that sample, what we've done is we've taken our radioactively labeled DNA that is our sort of suspected enhancer, and we've added this nuclear extract. So that source of proteins that we get by using the Downs homogenizer to break up the cells and get rid of the cytoplasm and the cell membrane and the ribosomes and so forth. But the pellet will have the nucleus, get that nucleus at the bottom of the centrifuge tube and burst it open and use those proteins, and we would have MyoD binding. And we don't know that yet, but a protein's binding, and we think it does from our earlier experiments. And when it does, and we run that on a gel, it moves slower because the MyoD makes it a bigger molecule. And so it's called a gel mobility shift assay. So now we can establish that we have conditions just in test tubes where we can get the MyoD to bind. Okay, I think I might have a slide of that. Okay, and without the protein, the DNA moves fast, and with the protein, it moves more slowly. So we get that mobility shift assay. Okay, next one. Now we know this DNA can bind a protein, and I call it MyoD, but in fact, we don't know what it is. So we'd like to study that protein. We've already shown from DNA deletion studies that this protein must be important for activating the target gene. We've shown that this is where it binds. And now we've shown that it does bind there in our experiments. And the next thing is to take this piece of DNA and take some beads. So some beads, like little pieces of dust, you can actually see them, like little pieces of sand. So take some beads and attach that DNA to the beads and put lots and lots of beads into a test tube with liquid. Have the bottom of the test tube be porous, which is a tube that holds the beads but lets liquid through, and then pass that nuclear extract over it. We know now that this mystery protein will bind, everything will flow through, everything else will flow through, and then we can release the mystery protein and study it. And we can make a great big one of these, and this tube is called a column because it looks like a cylinder. It looks like a house with columns. It looks like a big cylinder. And we'd work in the cold room. So there are refrigerated rooms in research lab buildings to do these kinds of experiments, keep things cold so we don't have any breakdown of our protein or anything like that. And then these columns can be great big. To purify protein, a person might use a column that is, 8 inches wide and 3 feet long. Yep, that big. And so our book shows that, again, chapter 7, and it shows this idea. We start out with these beads, and they have the piece of DNA that will bind our protein in it, and we put those in, and the beads are big enough that they can't flow through the porous bottom, but every small molecule could. So we put in our extract, and it includes, in this case, they're talking about a protein transcription called SB1. See, that's the 8th edition, and we're in the 9th edition, 7. And I'll get these updated, in our class going forward because I have the PowerPoints now. So we flow the liquids through, and this is called column chromatography. I guess you have some familiarity to that with from other chemistry and biology courses. So this is called affinity chromatography because we're taking advantage of the affinity of our protein for that DNA. We flow it through, and then later we release the protein, and we have our protein purified. So we can study it more. What is that protein? We're really super interested in it. Okay, so one more example of a method, and this method is different. This method is, instead of working in a test tube and breaking the cell open, we want to work in a more natural setting. So we're going to work in the cell, and in the living cell, we're going to take this and we are going to say we want to study, in our case, I think the example I have is we want to study HSF, and we want to find out what are its target genes. So this assay, which is called the chromatin immunoprecipitation assay, or CHIP, that's the spelling there. CHIP is used to identify all the target genes of a transcription factor. So this should serve to function to identify all of the heat shock genes, right? And I can tell you right now, there are 10 or 12 of them. Yeah. And we don't have them all. Maybe we have that first one. This could also be used to identify all the targets of MyoD. We know about myogenin, but it probably has other targets, and we want to know about them too, because they must be involved in muscles, right? So heat shock transcription factor, we've got a target gene. We identified and purified heat shock transcription factor. Now we want to identify all of them. And so here's how you do that. You start out with a living cell and just the cells alive. And what you need is a way to bind this transcription factor. So you have to take some of the purified transcription factor that you isolated with affinity purification, purified transcription factor, and you need to make an antibody. You need to have a protein that will stick to your protein of interest. And people draw antibodies that look like the letter Y because that's what the actual protein molecule looks like. And antibodies are part of the story of our immune system. They help us fight against infection. And so you would take an animal like a rabbit or a mouse, and you'd actually inject HSF into its blood, and the animal would make antibodies to it, and these antibodies would be used in your experiment. That's why it's called immunoprecipitation. And what you're going to do is you're going to use an antibody to pull down that HSF in a test tube after the experiment has been started. Okay. So the first step in this experiment is you would have in the cell all the DNA, and you'd have some heat shock genes that you don't know about yet, and you'd have that HSF bound to them. And the first thing you do is, in a living cell, you add a cross-linking agent. And a cross-linking agent is going to create random covalent bonds between molecules and all through the cell. So you do it briefly, and some of the HSF is going to be covalently cross-linked to the DNA that it's bound to. So in this case, of course, you have to use this cell after it's had a heat shock, so the HSF trimers are bound to the DNA of all the target genes. So that's the first step, and I'll just keep drawing on this surface here. So the second step is to lyse the cell. That means break the cell open and release everything inside. The third cell is to gently chop up the DNA into small pieces. The DNA is really long, and you gently chop it up. Instead of using an endonuclease, people actually usually just use a kind of like a blender, because the DNA is very long and very thin, And if you just blend it for a moment, it will get sheared or chopped, torn up into smaller pieces. And then the fourth thing is to that material you add your antibody. And then what we'll have in the tube is we'll have the HSF, and some of the HFS molecules will be have been cross-linked to some of the cis-regulatory regions for heat shock genes. And we'll just have a short piece here because we chopped up the DNA to short pieces. When we add the antibody, it will stick to this, and it will stick to all the HSF proteins. Most of the HSF proteins didn't get cross-linked because we just cross-linked for a moment. And some of the HSF proteins weren't even bound to DNA at the moment. But we can bind all the HSF, and whether it's bound to DNA or not, and it will also include HSF that's bound to every target gene. So not just this gene, but another gene, and all of them. And then, one way or the other, we immunoprecipitate. We pull that DNA antibody out of solution. Precipitation, is to mean to bring something out of solution. And we do that in a variety of ways. And a really common way is that this could be a magnetic bead that we attached before the experiment. And it will help us use a magnet to pull this out. And what we're pulling out is the antibody holding on to the HSF, which is covalently cross-linked to the piece of DNA. Then we'll reverse the cross-links so we can study now the DNA, and we'll sequence the DNA. All the DNA too, we'll sequence the DNA, and that means to read the letters. And in doing so, we'll discover everywhere that HSF was bound. So CHIP uses that. Start out and make an antibody to the protein you're trying to study. Use some of your affinity purified transcription factor to inject into an animal like a mouse or a rabbit. Make antibody. Use that antibody in this experiment. First step, cross-link a living cell that's been activated so that the HSF will be bound to heat shock genes, and just cross-link for a short time so we get some random cross-links, and then lyse the cell, gently chop up the DNA, add your antibody, pull down the HSF with an antibody, and it will drag along the cross-link DNA pieces that are the enhancers and cis-regulatory regions of the target, all the target genes. Reverse the cross-links, sequence the DNA of everything, and you'll find all the genes. Okay? So I have some slides that show that, and I think this is slides from this chapter as well in the 9th edition. I forgot to double check that, and it just describes that method. And we're done. Oh, no, let's just talk about this. So we found these heat shock transcription factors bind to the target genes of HSP genes, and they're just given names like HSP70, and we don't need to know those names. They're just, several small HSPs and so forth. And they're involved in protein folding and stability under stress conditions like heat shock. Then it says this: recent research has shown that heat shock transcription factors also regulate a wider range of genes involved in really interesting stuff, but depending on the situation. How did they study that? So on an exam, I should be able to say that there is a transcription factor, and it is known to bind to these regulatory regions and regulate expression of these target genes. Great. But now scientists know there are additional genes that do other jobs in other situations that are also regulated by that same transcription factor. How were those found? And the answer is CHIP, chromatin immunoprecipitation assay. And that's so important because usually you start out studying something like MyoD, and you're like, oh, I see MyoD is involved in activated myogenin. And you're like, so it's important in muscle development, yeah? And you're like, yes, it is. And then you're like, are there other muscle genes that just activated? And there are. Well, let's find out all the things that it acts on, right? Let's learn as much about it as we can. And then in that process of CHIP, you might find things like this, that, oh, wow, it's even actually involved in some other genes in other settings, not in muscle, but at other types, at other locations. Maybe MyoD does a different job, like that, because it has different target genes. And we're excited about that. What are the total target genes? So here it says identifying estrogen receptor target genes. We learned about vitellogenin, right? Vitellogenin, not present in us. I think there's only one gene, one L, one T, and two L's. Vitellogenin, yolk protein. And if we did CHIP in a chicken, it would pull down vitellogenin and all the other genes in a chicken that are important for adolescent development in females. So see, it just says estrogen receptors are ligand-inducible transcription factor that regulates a large number of target genes. These targets are particularly relevant in breast cancer. What kind of breast cancer? ER-positive breast cancer, right? That the sensitivity of the tumor to estrogens determines whether the patients can be treated with endocrine therapy. Right? So we understand that and should be able to express that knowledge on a test. Identifying genomic estrogen receptor targets is a daunting task. Quantifying expression levels of suspected target genes after simulation or using experiments, this effect will reveal that. So we're going to get down here. However, the estrogen receptor can bind to a variety of different response elements, so not even the ones that are most familiar. We use CHIP to identify all target genes. Woohoo! Right? So good stuff. All right, and with that, I bring in vitellogenin again, and we're done with really the core of Chapter 7 that I want to talk about, but I want to answer lots of questions as well. So, Snow Day lecture on to Chapter 16 and receptors next week. Yep, all right, great. Thanks a lot.